dear friends, how are you all? After a long gap, I'm back with a new topic. Actually, this is a continuation of the previous topic. Today, we will mention about the remaining portion of reproduction in flowering plants. That's detail about the female reproductive part. So let's start with the reproduction in flowering plants, female reproductive part. Female reproductive part is the Genetium, and they are having three main parts like style, stigma and ovary. Stigma is a terminal portion which is very sticky, and the receptive part, why we call them as a receptive part, because they there is region attract the pollen grain for pollination. Then comes the style, which is the elongated part connecting the stigma and the ovary. This style carries the pollen tube to the ovary for further fertilization process. Now, ovary is a basal swollen portion where the fertilization process is happening, and this portion produces the female gametes. So, obviously, we know fertilization process will be carried out. Clear? Next, let us study about the detailed portion of the ovary. Ovary, there is actually it's an oval in shape, structure, and there ovary is having an empty space or cavity which is called as locule. This locule is having a parenchymatous cushion-like structure, that is parenchymatous tissue structure, that is called as placenta. And in this placenta, the ovule is attached. Ovule is a main portion of the ovary which carry the female or which produce the female gamete. If you go into the structure of ovule, ovule is surrounded by two integuments, that is a two protective layer. And this ovule is attached to the wall of the ovary with a stalk that is called as funicle. Now the point of attachment of funicle to the ovule is called as hilum. Just look into the picture and make it clear. Okay. Just look into the picture. There you can see in the ovule an opening, right? Opening where the integuments are opened. The opening region is called as micropolar end of an ovule, and that opening is for the entry of pollen tube into the ovule. The opposite side of the ovule, where it is completely closed and from where which the integument arise, that portion is called as chalaza. Clear? Inside the ovule, that is inside the integument, there are mass of cells that is called as nucellus. So that is the structure of an ovule, very important. Normally question will come for the exam. Structure and function of an ovule. Okay, next we will go to the process of development of nucellus, that is called as Megasporogenesis. Megasporogenesis is a formation of megaspore from megaspore mother cell of ovule, that is the nucellus cell of the ovule. Here, one of the nucellus cell, which will be called as megaspore mother cell, undergo meiotic division. We know meiotic division happens in two stages, meiotic 1 and meiotic 2. At first division, the microspore mother cell is divided into two daughter cells, and then again they undergo meiotic 2 division, and as a result 4 daughter cells are formed. That is 4 megaspores are formed in a tetrad form. Out of this 4, 3 megaspore at the micropolar end will get degenerated. So they are not functionable. Only one at the chalazal end become functional. Okay, this is entirely different from that of microsporogenesis of male reproductive part. Let us see the development of this megaspore, that is called as megagametogenesis, that is a formation of gametes from the megaspore. Now how it happens? Here the functional megaspore, that is only one megaspore is functional, right? This megaspore undergo free nuclear mitotic division. It is not meiotic division, it is mitotic division. Please note that. And here they undergo 3 successive mitotic division, and as a result you can see in the picture how is the nuclear division happening. First of all, the megaspore undergo first mitotic division. As a result, 2 nuclei are formed. Again, these 2 nuclei will move into 2 opposite direction, and they undergo second mitotic division and they form 4 nucleus. Then again they undergo third mitotic division, and as a result in 2 parts they form altogether 8 nuclei. Clear? That is 4 plus 4 nuclei are formed at the 2 direction, that is at the chalazal end and the micropolar end. Now 1 nuclei from chalazal end and one from the micropylar end move towards the center. And this is the arrangement: 3 at the micropylar end, 3 at the cella cell end, and 2 which is moved towards the center. Finally, the wall formation take place and daughter cells are differentiated. So how is a daughter cell formed? After nuclear division, we know cytokinesis happen. That is, wall formation, cell wall formation happens, and as a result daughter cells will be formed. Here, how is a daughter cells formed? At the cella cell end, there are 3 nuclei, and as a result they form 3 daughter cells, that is 3 antipodal cells. Next, at the micropylar end, there are 3 nucleus, and out of this 2 nuclei form 2 synergids, and the 1 nuclei they form into egg cell. So in the micropylar end there are 3 cells, that is 2 synergids and 1 egg cell. At the center already we have told 2 nuclei have moved from the cella cell end and the micropylar end. These 2 nuclei are called as the polar nuclei. 2 polar nuclei together forms a central cell. So the ovule structure finally after division with the daughter cells are called as embryo sac. So we can say an embryo sac is having many cells with multinucleated structure. So we can say a mature embryo sac is a 7-celled 8-nucleated structure. Just look how many cells are there and how many nucleus are there. Now, there are 3 different types of embryo sac development which you have to study. The first one is monosporic type. Here it's a normal type of division, which already we have mentioned how the embryo sac is formed. Embryo sac is formed from a uninucleated megaspore. That is a normal type which we have seen now. Second type is bisphoric type. It's little bit different. Here the embryo sac is developed from a binucleated megaspore. So there will be 2 nucleus inside a megaspore and they divide to form an 8-nucleated 7-celled structure. Now how it happened? One of the megaspore mother cell undergo first meiotic division and they form a dyad. In this dyad, one of the daughter cell get degenerated and only one become functional. This functional cell again undergo second meiotic division and they form a binucleated structure. Now this binucleated megaspore undergo a normal mitotic division 1 and 2, and as a result a normal embryo sac will be formed with a 7-celled and 8-nucleated structure. Next, let us study another type of embryo sac development, that is tetrasporic embryo sac development. Here 4 nucleated megaspore undergo the division and as a result 8 nucleated or 16 nucleated embryo sac will be formed. Now you will wonder how this will happen, because obviously a megaspore is having only a single nucleus. Now let us see how it happened. Here the single megaspore mother cell undergo first meiotic division and second meiotic division, and there 8 nucleated megaspore is formed. Here there is no formation of dyad or tetrad. There is no cell wall formation, only nucleus is divided and 4 nuclei is formed in a single megaspore. Now the single megaspore with 4 nucleus undergo mitotic division. In some plants they will undergo only 1 mitotic division. Here there is no 3 successive mitotic division as which is happening in the normal embryo sac development. In some plants only 1 mitotic division will happen. As a result, they will form an 8-nucleated embryo sac. Whereas in some other plants, they will undergo 2 successive mitotic divisions, and as a result, an 16-nucleated embryo sac will be formed. Clear? Okay, so that is all about the female reproductive part of a flower, which helps for the sexual reproduction in plant. Okay, so thank you once again for watching my video. If you like my video, please like, share and subscribe and support me. You wait for me for the next video to come as soon as possible. Okay, thank you once again. Have a nice day. May God bless you all. Bye bye. See you.